So this is a fun problem, especially for people that have had Labrador Retriever dogs. That they uh, most likely questioned what was the dominant fur coat color in Labrador Retrievers, and so this problem actually addresses that and looks at the system of fur coat color and how it's inherited in black labs. Okay, so it's 426. It says multiple crosses were made between true breeding lines of black and yellow Labrador retrievers. All the F1 progeny were black. When these progeny were intercrossed, they produced an F2 generation consisting of 91 black puppies or 39 yellow labs and 30 chocolate. So one, it wants you to propose it wants you to propose an explanation for the inheritance of coat color in Labrador retrievers, and B, it wants you to propose a mechanism, a biochemical pathway for coat color determination, and indicate how the relevant genes control coat color. Okay, so in this one, just like other problems, we want to sort of summarize what it's telling us. And so here, I wrote 422. I'm not sure why this is problem 426, but in the parental generation, you have a black dog mating with a yellow dog, right? Or black dogs versus yellow dogs, just because you have so many of the F2 here, we're assuming that there's multiple breedings, right? Um, but both of these come from parental populations. So parental populations mean that they're true breeding black dogs and true breeding yellow dogs. When you mate black with yellow, you get black out of that. So that's instantly telling you in the F1s that the black phenotype is dominant. You then mate black and black, right? And if this were a monohybrid cross, a classic monohybrid cross with one gene, two alleles, dominant recessive interaction between the alleles, like we saw in lots of Mendel's, in lots of cases with Mendel's monohybrid crosses, we would get a 3-to-1 ratio out of this F1 cross, black to yellow, right? But we don't. Instead, in the F2 generation, we get another phenotype coming out of that, and that's the chocolate phenotype, the brown lab puppies, and we get 91 black, 39 yellow, and 30 chocolate. So if we look at these numbers, we can see that they pretty closely approximate. We don't really need to do a chi-square test, right? Because there's 9. This is a very, very close approximate to a 9:4:3 ratio. So now that we have that 9:4:3 ratio, again reminding you that the 4 is a combination of the 3 and 1 ratio from the 9:3:3:1 ratio, we instantly know that there are two genes. There's two alleles in each of these genes. Those alleles are interacting in a dominant recessive fashion, and the two genes are independently assorting. And because it's this combination, we know that some sort of epistasis is going on. And so just from the ratios of those F2s, we instantly know that. We've also figured out that the black phenotype is dominant to the yellow phenotype. Okay, so that answers part A for us. Now we have to propose a biochemical pathway. But first, let's go back through and say, okay, now we know there's two genes. We know the interactions. Let's go through and generate the genotypes for each of these generations, just so that we can be really comfortable with what's going on in the system. And so here, we would say, okay, a true breeding black population is going to be homozygous dominant for both of those genes. A true breeding yellow population is going to be homozygous recessive. Why? Because here we see, well, there's a couple of reasons, but here, we obviously see that the black is the 9 part of the 9:3:1 ratio. That's the dominant, right? And then the yellow is the 1 part, and that's usually the recessive, right? The recessive homozygous alleles in each of those genes. Okay, so then we mate those two together, and we get an F1s that are black, right? Classic dihybrid cross, right? So you have a big A allele, a big B allele, you have a black phenotype, and then you cross that with another big A, little a, big B, little b, and you get the potential genotypes, and we can just draw them out here. A, big A dash, big B dash, big A dash, little b, little a, big B dash, and little a, little b, little b. So for these phenotypes, we could already see from up here that the A dash, B dash, the big A dash, big B dash is going to be the 9 part of that ratio, right? we can see that the little a, little b is going to be the one part. It's going to be yellow, right? That's the yellow one. And then we just assign the other three to the 2, 3 parts of the 9-3-3-1 ratio to big A dash little b, the dominant recessive, and then the recessive and the dominant. Okay, so now we have these genotypes. So we have to figure out, okay, what would be the epistatic pathway? And so just like we usually do, we draw out these in the cases of epistasis where we have 2 genes, we usually draw these 2 arrows out first. Here's one arrow, here's another arrow. Usually on this side is the dominant phenotype, right? Usually on this side is the recessive phenotype. And so we place the black over here, the yellow over here. Now all we have to figure out is, you know, which gene causes the yellow to go to chocolate. The chocolate goes here because it's the only one that's remaining. And so what you see is the big A causes you to go from yellow to chocolate. If you're chocolate and at the other gene, you have little b, you stay here at chocolate, right? So here you have the big A dash that brought it to chocolate, little b stays at chocolate. But then if you have the big B allele here, you then go from chocolate to black. And that's our epistatic pathway. And so in this case, the little a alleles, right, are epistatic over whatever's at the B. So if you have a little a, you're going to override whatever happens downstream here at the B gene. And so here, it matters what's happening here. Once we have what's happening here, that's going to that well, not what's happening, but if you have the little a here, it's just going to stop it at the yellow. And so there you have it. This is the 2 gene system that that shows the inheritance of fur coat color in Labrador retrievers. Okay, let's stop this. See if I can